All right, let's look at the stages of meiosis. So on the top here for all of these is a microscope image of chromosomes, and they've been the cells have been smashed, so they're kind of spread out a little bit so we could see them. Normally they would be pretty compact. And on the bottom is a cartoony version of what is happening here. Okay, so prophase 1, this is where crossing over occurs. So prophase 1 is really long because all the chromosomes have to condense, just like they would in prophase of mitosis. And if you remember from mitosis, prophase of mitosis is also really long. So you can see this kind of braided appearance here. That is the chromosomes crossing over. And notice there isn't just one crossover location, there's multiple. So 1, 2, 3, 4, 5 on this one chromosome here. And there's each chromosome has 2 identical sister chromatids. So what you're actually seeing here in each one of these little crossing over chromosomes, there's actually 4 chromatids there. So each chromosome has 2 chromatids, and there's 2 homologous chromosomes synapsing together. So we have 4 chromatids synapse together. So during prophase, the centrioles have replicated already in G2. DNA replication occurs in interphase, in S phase of interphase, just like it does before mitosis. So that's already occurred. So prophase, we have condensation, crossing over. Metaphase, still similar to mitosis in that those centromeres are going to line up on this kind of imaginary plane, the metaphase plate. But the 4 chromatids are still stuck together. So we call those tetrads, tetra for 4. So those tetrads are stuck together. We have a spindle formed made out of microtubules, just like before, just like in mitosis, except now the 2 sister chromatids are not going to separate during anaphase. What happens during the first division is the separation of the homologous pairs. The homologous pairs separate in that first division. So each chromosome here still has 2 sister chromatids. They're not identical anymore because of crossing over, but they still have 2 chromatids. So they are separating and going to the poles. So you notice the Roman numerals here, 1. That's the first division. And don't use a Arabic numeral 1. We use the Roman numeral 1 to represent that. Okay, telophase 1, nuclear membrane reforms. And there really isn't an interphase in between the 2 divisions. The chromosomes decondense a little bit, but not really. They don't really go back to interphase. So here we have telophase. And now we're not going to have another round of DNA replication. DNA is not going to replicate in between meiosis 1 and meiosis 2. It doesn't have to because the chromosomes still have 2 sister chromatids after this first round. Each cell now has half the number of chromosomes though. It's this first division that's what we call the reduction division. All right, so now we have 2 cells. They have half the number of chromosomes. So back here we had 4 chromosomes. Now each cell has 2 chromosomes. So that's our reduction division. They are already haploid with only 2 chromosomes. The sister chromatids are no longer identical, however, because of crossing over. So no real interphase here. There's no S phase. There's no DNA replication. However, the centrioles will have to replicate again if it's a species that has centrioles. Those will replicate again, getting ready for prophase 2. So prophase, or meiosis 2, is just like mitosis. We now have single chromosomes with 2 identical sister chromatids, and those sister chromatids are going to separate. So just like mitosis, they're going to line up at the metaphase plate. They're going to separate during anaphase, and we're going to end up with 4 cells, 4 daughter cells that are all different. And now the reason that they're all different is because these sister chromatids are no longer identical, because one of those chromatids crossed over with the other homologous chromosome and swapped some DNA. So that's how we end up with 4 daughter cells that are all different, because some of the chromatids are what we call parental type, and they didn't cross over, and some of the chromosomes are recombinant chromosomes that have bits from both the maternal and the paternal chromosomes from the original homologous pair. So there's our daughter cells, our 4 daughter cells that are all different from each other. They are haploid, and each chromosome only has 1 chromatid. So here are, again, prophase 1 and metaphase 1. These are the 2 stages where the weird stuff happens. The first division is the one that's weird. So prophase 1 is really long. It's over 90% of the total time for meiosis, just like in mitosis. Prophase as well is close to 90% of the total time required for mitosis. So synapsis occurs during prophase 1. Synapsis means the homologous chromosomes line up gene by gene, and 2 of the non-sister chromatids are going to cross over. Metaphase 1, this is where independent assortment occurs. So metaphase 1, all of those tetrads are lining up on the metaphase plate, and which way those chromosomes are going to go is random. So that's going to increase the number of potential gametes that one individual can produce. And we'll go through the math on that, how many possible different gametes an individual can produce. But it's very much similar to mitosis in that there are kinetochore complexes on the centromeres that are going to chew their way up the microtubules, except in this first division, we're going to separate the homologous pairs, not the individual sister chromatids, not yet. There's something slightly different that happens during sperm formation and egg formation. For sperm formation, we have meiosis here, and we end up with these 4 haploid cells. All of those cells are going to go on to form sperm. They do have to change quite a bit, though. They're going to cast off almost all of their cytoplasm. The nuclear material is going to condense, and sperm are very tiny compared to a typical cell. And they're essentially a nucleus that can swim. They really don't have much else. They barely have any mitochondria. They do have some mitochondria that can make ATP for the flagellar swimming. But they can't really do any other kinds of metabolic activity or gene expression. They're a delivery mechanism for a haploid nucleus. The egg, on the other hand, has to have all the nutrients for the embryo to survive for about 2 weeks. That's how long it takes for, well, in humans anyway, that's about how long it takes for that fertilized egg, then developing embryo, to start to get nutrients from the mother. It takes a couple of weeks. So the egg has to be packed with nutrients. And a typical human cell is not nearly big enough. So the oocyte is actually the largest human cell in diameter. And it gets that way partly by asymmetrical divisions. So the first meiotic division and the second meiotic division occur, but they're very asymmetrical. And the first one that occurs, this one occurs during when the oocyte is preparing to be ovulated. So prophase here begins in all of the oocytes. Prophase 1, for mammals, it actually starts during fetal stages for the female. So for humans, it starts at about when a human female is about a 7-month fetus. That's when all of the oocytes in the ovary are starting meiosis, and they get stuck in prophase 1, and they sit there in prophase 1. Then when that egg is getting ready to be ovulated, the first meiotic division finishes. And we get this little polar body that is it doesn't have any function. It's just a garbage bag for the extra DNA so that we can have a haploid cell. The second division, reducing this nucleus now to the mature egg nucleus, doesn't occur in mammals until fertilization actually occurs. So slightly different process, but so we have our oocyte here that's going to go through meiosis. We only end up with one egg from that. So all the cytoplasm ends up with one cell, and the difference in size is even more disparate than it looks like here. The polar bodies are absolutely tiny. You will not see them looking through the microscope unless you're really looking for them and are using perfect cell staining technique. They're very hard to find because they're super tiny. But what we end up with then is a cell that is massive for a human cell and is packed with all kinds of goodies, including RNAs and fatty acids and ribosomes and mitochondria, and everything that will allow that little developing embryo to survive for several weeks until, well, 2 weeks, until implantation occurs and the embryo can start getting nutrients from the mother. So again, meiosis has some unique features. One is synapsis and crossing over. Synapsis is the chromosomes coming together, and crossing over is the recombination of two of the non-sister chromatids. So of those 4 chromatids that are lined up, 2 of them are not going to cross over, and 2 of them are. So we're going to have 2 chromatids that are going to stay parental type, and 2 chromatids that are going to be recombinant chromatids. And that's how we're going to end up with 4 daughter cells that are all different. And we call them daughter cells whether they're going to form sperm or eggs or some other kind of gamete. And so it's that first division that is the reduction division. The second division of meiosis is just like mitosis. I include this in your PowerPoint because it's a really nice summary comparing the differences between mitosis and meiosis. So we see here meiosis 1 is the one that's different. This is the reduction division. This is the division that reduces the chromosome number in half. The second division of meiosis is just like mitosis. We separate the sister chromatids, but because there's 2 divisions here, we're going to end up with 4 daughter cells. And because the sister chromatids are not identical, thanks to crossing over, we're going to have 4 daughter cells that are all. But there are mechanisms that we've already talked about in this chapter that increase genetic variation in offspring when the species has sexual reproduction. So if the species does not have sexual reproduction, like for example, amoeba that only divide by mitosis, all the offspring are going to be identical genetic clones except for new mutations. In species that have sexual reproduction, like animals, most plants, fungi, most protists, we have additional factors that are going to increase the genetic variation among the offspring so that all the offspring are going to be genetically unique individuals. So besides new mutations that are occurring each generation, we also have the independent assortment of the chromosomes during meiosis. So when the gametes form, the maternal and paternal chromosomes from the previous generation randomly go to the daughter cell. So we can have a huge number of different gametes just based on that. And we'll go over some numbers for that. Crossing over, that's the 2 non-sister chromatids for a pair of homologous chromosomes exchange parts of their DNA, creating new chromatids that did not exist in either of the parental generations. And then we also have random fertilization, which we know fertilization isn't entirely random, but the combination of when you have 2 different parents, every offspring is going to have 2 different assortments of chromatids from the 2 parents. in a sexually reproducing species. Okay, so if we just look at how independent assortment of the chromosomes increases genetic diversity. So here we have our species that has 3 pairs of chromosomes. So that means the gametes each have 3, the N number is 3, and then the diploid zygote is going to have 6 or 3 pairs. So, and then this cell, this diploid zygote, or the diploid cell of the adult, is going to go through meiosis. And when the gametes form now, we have the maternal and the paternal chromosomes are different colors here. If it's a diploid species that has 3 pairs of chromosomes, that means we have 8 possible gametes that can be formed, 8 possible haploid gametes with different combinations of the maternal and paternal chromosomes. So what about for species that have more than 6 chromosomes, which is most species? So the formula for figuring out the total number of different gametes that are possible just based on independent assortment of the chromosomes. We're not even going to count crossing over yet. The formula is 2 to the n, and n, remember, is the number of unique chromosomes. So for this species, that n number is the haploid number of 3. So 2 to the third is 8. So 8 different possible combinations. Now for humans, the n number is 23. So 2 to the 23rd is about 8.4 million. So that means that for each individual human can produce about 8.4 million different gametes just based on combinations of maternal and paternal chromosomes. That's not even counting crossing over, which is going to increase that even more because half of the chromatids that are involved in meiosis are not going to be the paternal types. They're going to be recombinations of the 2 different ones. So more than 8 million different possible gametes just from independent assortment. Now crossing over is going to increase that even more. So let's look again at crossing over. So here we have our sex cell that's going through meiosis. It's in prophase 1 of meiosis, and prophase 1 is when we have synapsis of the pairs of homologous chromosomes, and then crossing over is going to occur. But all the chromatids are not crossing over, only 2 of them. So 2 of the chromatids are going to stay parental type, and 2 of them are going to cross over. So 2 of the non-sister chromatids are going to cross over, and you'll notice they call this crossover point a chiasma from the Greek letter chi for X. So, because scientists like to name things, and this just means X-shaped. I'm not going to ask you for that word on the test. So this little chromosome here that they're showing in this TEM image just has 2 crossover hotspots, but as we saw earlier, a longer chromosome might have up to 5 or 6 different crossover hotspots, or crossover spots rather. For vertebrates, there are crossover hotspots as well, which we'll talk about. So what we end up with is our pair of homologous chromosomes. We have 2 chromatids, which are what we would call parental type, meaning they didn't recombine, and we could trace back this exact chromatid to one of the parents. The same here. We can trace back this chromatid to one of the parents. And now we have 2 chromatids that are recombinant chromatids, meaning they have recombined the DNA from 2 different parents. So we get, because of recombination, we get new combinations on a single chromatid. Now, why would this matter? If the genes are the same, why does it matter if they're 2 different alleles are next to each other now that weren't before? Well, one reason is because there are control regions for these genes. So when you have a recombinant chromatid like this, the gene may be the same, but the control region for that gene might now be from the other parent of that individual. So you could have the same protein being produced, but at different levels. So here's anaphase 2, the second division, just like mitosis. And so now we're going to end up with 4 daughter cells. This is how we get 4 daughter cells that are all different because of crossing over, because we have 2 chromatids here that are not parental type. And so we, in fact, for every pair of homologous chromosomes, this occurs. So random fertilization increases that. So if we consider for humans that each person can produce about 8.4 million possible gametes just based on independent assortment, not even counting crossing over, and we would have to at least double that for crossing over. That means that each individual can produce about 70 trillion different diploid combinations. So for any 2 people who are reproducing, we can produce up to 70 trillion different diploid combinations. And that explains why even in families where there are a large number of siblings, none of them are going to be genetically identical, because there's just way, way, way too many possible combinations. Now, you probably do know that there are humans that are genetically identical, but they didn't come from 2 separate fertilization events. So for species that have sexual reproduction, what all this means is that each zygote is going to have a unique genetic identity. And this must be a huge advantage in evolutionary terms, because it has been selected for by natural selection and maintained by natural selection. The exceptions are the ones that no longer have sexual reproduction, like there's a few animals, there's a few protists like amoeba that don't have sexual reproduction, but by and large, all eukaryotes have sexual reproduction. So it must have a huge advantage. What could be the possible disadvantage? Well, if you have an individual who's a particularly good genetic combination, that exact genetic combination is not going to occur again. That's the downside of sexual reproduction. So species that do both, like Hydra, that have both sexual reproduction and vegetative reproduction, or reproduction by asexual means, if there's a particularly good genetic combination for one individual that's very survivable in a particular environment, that individual can also reproduce only by mitosis asexually. But vertebrates, for the most part, don't do that. There are genetically identical individuals, though, identical twins. And identical twins are natural genetic clones. We call them monozygotic twins, meaning mono means one, zygote is zygote. So monozygotic means they're from a single zygote. So that means one egg, one sperm, one zygote. And at some point later in development, it splits. Sometimes that split is incomplete, and that's how we get conjoined twins, which luckily is extremely rare. Almost all identical twins completely separate in the embryonic stages and form 2 completely separate embryos. Interestingly, we don't know what causes the embryo to split, but it's not genetic, because identical twins don't run in families. So if a woman is an identical twin, or if a man is an identical twin, they have no greater chance of having offspring that are identical twins than the general population. So that tells us that that it's not genetic. So if it was genetic, that would mean that individuals who were an identical twin would have a greater chance of having children who are identical twins, and that's not the case. So that tells us it's something environmental. We have no idea what that might be. We do know that when in vitro fertilization is used, the chances of identical twins increases. So handling the embryos outside of the body increases the chance that there will be identical twins produced. But it does not run in families. Now, there's always somebody who says, well, I have an aunt who's an identical twin, and then she had children or grandchildren who are identical twins. Yeah, that's going to happen occasionally. If identical twins are completely randomly occurring in the population, occasionally there'll be multiple sets of identical twins in one family. But when you analyze population data, it indicates that identicals they do not run in families, so it's not genetic. The most identicals that have ever been born and survived, or that we even know of, are the Dionne Quintuplets, who were born in 1934. There were 5 identical girls, and there was actually a 6th identical who died during the pregnancy and was not born alive. They're the only identical quints known to be born alive. Of course, it's quite recent. There could have been, humans have been around for 250,000 years. It's quite possible that there were multiples that were born in the distant past, and there would be absolutely no record of them, and they would have no chance of surviving, because of course multiples tend to be premature. And it wasn't until the last 100 years or so, when oxygen became available and the type of neonatal intensive care that we have now, that premature infants would have much of a chance to survive. So the more identical twins are really common. Probably everybody in the class, you probably have at some point in your life had a classmate who is an identical twin, or identical twins in your neighborhood. It's fairly common. Identical triplets, much more rare. In my life, I have only known 1 set of identical triplets in my life. They went to my high school. Identical quadruplets, much more rare. There's only 70 sets approximately of identical quadruplets known in the world right now, so that's quite rare. Identical quints, I don't know if there have been other sets of identical quints. There's no record of any surviving, or identical sextuplets or more. There's no record of those. But of course, such high multiples would have only been able to survive just in the last 100 years or so. Some of the Dionne quintuplets are still alive. The last time I checked, I think 3 of them were still alive. They would be in their 80s now. But this is, again, we don't know what caused it. The woman who gave birth to them was not on any medications of any kind. They had 1 sister who was a singleton birth. So we have no idea what caused it, but it's not genetic. Now, multiples who are not identical, the most common would be, of course, twins. So we would call non-identical twins, which are commonly called fraternal twins, we would call them dizygotic twins. And they are not any more alike genetically than any other kind of sibling. So because they're from 2 completely different fertilization events, 2 zygotes, so that's why it's di. Di is Greek for 2, bi is Latin for 2. So dizygotic twins means 2 zygotes. So they develop completely separately. Dizygotic twins do run in families. So in the female side, because it has to do with multiple ovulations, most women only ovulate 1 egg per month. But some women ovulate from both ovaries every month, and that is genetically controlled. So if a woman is a dizygotic twin, she has a much higher chance of giving birth to dizygotic twins than the general population. So that one does run in families. Fraternal twins do run in families, and it is genetically controlled. But it can happen randomly as well. Sometimes it can happen to somebody who has no record of any twins being born in their family. So dizygotic twins, just like identical twins, sometimes don't separate completely and we get conjoined twins. Dizygotic twins can also fuse during early embryonic stages, and they will form one normal-looking baby who at birth looks like a completely normal singleton birth, but is in fact a mixture of cells from 2 different individuals. And we call these individuals chimeras. We have no idea how common this is, because the individuals who are chimeras are usually only discovered accidentally. The one that I first remember was about 15 years ago or so. There was a family where the daughter had cancer and needed a bone marrow transplant. And they did have other younger kids in the family. And of course, the parents get tested as well to be donors, because that would be the ideal, of course, if they're bone marrow donors, that somebody in the immediate family can be a bone marrow donor. So they tested everybody. to see who would be a match. And then they called the parents and said, come on into the office and we'll give you the results of your tissue typing match. And when the parents arrived, the doctor had called the police because the test results showed that the mother was not the mother of the children, but was in fact their aunt. So only sharing 1/4 of her DNA with her children rather than half, as we would expect for the mother. And she said, you know, I have witnesses. I gave birth to these children. They looked into her medical records, and sure enough, she had given birth to those children. So the doctors then took tissue biopsies from all over her body, and it turned out that she was a chimera of 2 female non-identical twins who had fused. And so her ovaries and her uterus were from one twin, and her bones and so therefore her blood, which they were taking a sample of, was from the other twin. So her bone marrow was, when they did a sample of her blood, they got the one twin, but in fact the egg came from the other twin. So, and we since now know of dozens of individuals like this who are chimeras. Now, you can also have a chimera of a male and a female dizygotic twin. And we have discovered individuals who are chimeras of a male and a female twin. In this case, the individual will be most likely look like a normal phenotypic male at birth, and could even be fertile if the male twin is the tissue that the testes were formed from. But sometimes it's a mixture. Sometimes they have only one normal testes and the other one is doesn't develop correctly because it's actually tissue from the female twin. So luckily as well, just like conjoined twins, chimeras are rare. Now you can have fraternal siblings who are more than 2. That's less common as well, just like for identicals, but it's more common than the identical triplets, identical quadruplets, which are extremely rare. Fraternal triplets, fraternal quadruplets are much more common, especially because they can result from fertility treatments and from in vitro fertilization. The most that have ever survived are octuplets, and there are several sets of octuplets now that have survived, although they almost always have complications because they're going to be extremely premature. An 8-baby pregnancy is an extremely high-risk pregnancy. There have been documented pregnancies of more than 8 babies, and none of those babies has survived at all, not a single one. They always miscarry too early for any of the babies to survive. And the same is true for most multiples above triplets. So quadruplets and above, those pregnancies, unless it's not the first pregnancy for the woman, if it's her first pregnancy, usually the babies will be born way too early, even for our really good neonatal care that we have today, and none of them will survive at all. So even a twin pregnancy is considered very high risk because of the risk of very premature delivery, especially if it's the mom's first baby. This is Natalie Suleiman. She's probably the most famous individual who has multiple babies. Her doctor actually lost his medical license because of implanting 8 embryos into her. Most states now have laws regulating how many embryos can be implanted just to avoid a situation like this, which is very dangerous both for the babies and for the mother. So I mentioned earlier that sexual reproduction must have a very high advantage in evolutionary terms. So remember, things that are selected for by natural selection are things that make reproduction and survival more likely in a particular environment. So sexual reproduction evolved at least 2 billion years ago, and it's been maintained ever since, and almost all eukaryotes have sexual reproduction at least sometimes in certain environmental conditions. And some exclusively have sexual reproduction, like vertebrates. Almost all vertebrates have sexual reproduction as their only means of reproduction. So why? What is the advantage of sexual reproduction? Because there is a distinct disadvantage that I mentioned, that if an individual is particularly well adapted to their environment genetically, if it's a species that only has sexual reproduction, that exact genetic combination is not going to exist again. It's going to be lost in the next generation. Their alleles will be passed on, but not their exact genetic composition is not going to be passed on. So the advantage has to be in the variation, that having offspring that are varied has to be an advantage in itself. And think about changing environments and changing environmental conditions that have occurred over the last billions of years here during evolution, that producing offspring that are not identical, producing offspring that are varied in their genetic traits, has been an advantage for most species, such an advantage that it has maintained sexual reproduction despite the risks. For many species, there is high risk associated with sexual reproduction, but the advantage overwhelms that risk. For vertebrates, not reproducing sexually is in fact very, very rare. This is a reptile species that is one of those rare exceptions. They lay eggs like most lizards do, and the species is entirely composed of females. Initially, when that was discovered, it was thought that they just weren't identifying the males, that the males were maybe looked like another species and had been misidentified. But in fact, we now know that this species of lizard, which lives in the western United States, has only females. Meiosis doesn't occur. The eggs are diploid, the adults are diploid, there's no recombination. So when a female lays eggs, all of the offspring are going to be identical clones of herself. And the reason the species is all female is because it's the females that lay the eggs. Males would not, a species that was all males would not have any way to reproduce that way. This type of reproduction is sometimes called parthenogenesis, and there's a few, occasionally this can happen. There's occasionally snakes in captivity that have no mate. Female snakes will occasionally lay fertile eggs, and in that case, again, they're clones of herself. It's rare, but it's not so rare that we don't see it occasionally on a regular basis. So how did sexual reproduction evolve from those early eukaryotes that would not have had sexual reproduction? So we talked about the evolution of the eukaryotic cell, that first we had the evolution of nuclear membrane, and then bacteria was engulfed that later became the mitochondria. So at some point, this forming of haploid cells and those recombining and then forming a zygote and meiosis, that evolved at some point. And interestingly, meiosis uses some of the same enzymes that are used for double-strand DNA repair, and DNA repair occurs during meiosis as well. Double-strand DNA repair occurs. So it's quite possible that these two things are more related than they appear, and that meiosis actually evolved as a direct result of evolution of these DNA repair mechanisms. And crossing over as well became possible because of the evolution of these DNA repair mechanisms. Another hypothesis is that recombination reduces negative mutations or deleterious mutations in a population. This hypothesis is called Muller's ratchet, and the idea is that if you have an organism that's producing thousands of offspring, some of those offspring are going to get a higher percentage of the deleterious alleles, other offspring are going to get fewer. The ones that get fewer of the deleterious alleles are the ones that are going to survive. So it's a way of getting rid of deleterious mutations in a population. Another hypothesis is that the evolution of diploid organisms, which doesn't exist in prokaryotes, diploidy allows every individual to have at least 2 copies of each gene. So if one of those copies is bad, that's okay. And it's a way of storing these kind of alternative versions of a gene that might be bad in one environment, but if the environment changes, that mutated allele might actually turn out to be an advantage. So diploidy itself has been selected for as an advantage as well. So in meiosis, we talked about, oh, and you have to look at the cute little skunks up here with their siblings that are not identical, one with a little different fur color. So meiosis, and again, we can tell that this is the type of cell division for producing eggs and sperm because there's an egg here and a little sperm right in the word. It's a type of cell division that produces gametes, which could be eggs or sperm. In plants, that would be pollen and ovules. And protists, they could be the same size, and it could be just plus and minus A and alpha gametes. We reduce the chromosome number by half so that when syngamy occurs or fertilization, that we restore the chromosome number. We have two processes that occur that increase genetic diversity: synapsis and crossing over, and also independent assortment of the chromosomes. Meiosis results in 4 daughter cells that are all different. They're different from each other because of crossing over, and they're different from the mother cell in that they have half the number of chromosomes. Sexual reproduction and meiosis are advantageous, and most eukaryotes have sexual reproduction and meiosis at least occasionally, and many have asexual reproduction as well. So it's that first meiotic division that is the weird one. Prophase 1, that's when we have synapsis of the homologous chromosomes and crossing over. That first metaphase, we have the tetrads lining up on the metaphase plate, and the homologous chromosomes separate. Each homologous chromosome after that first cell division, though, still has 2 chromatids. But now we have haploid cells. So if you look up at this first cell here, we have 6 chromosomes. Each of the daughter cells here after the first meiotic division only has 3 chromosomes, so 1 copy of each one. And we have some chromatids here that did not exist in the parental cells.